Dear students, we are continuing in this session the molecular basis of inheritance. In the previous classes, we discussed the nature of a genetic material, that is DNA, how it was proved. Subsequently, the structure of a DNA was elucidated or discovered by Watson and Crick. That was a landmark point in the history of molecular biology or science. Then question came, DNA, the genetic material, how it replicates. The most thoroughly studied organism in this earth is E. coli, Escherichia coli, which has a single circular DNA as its chromosome, but what is the length of that chromosome? Similarly, if we come to eukaryotic cells, the number of chromosomes varies from species to species. We have 46 chromosomes. The pea plant, the garden pea has 2n is equal to 14. Likewise, different eukaryotic organisms have different number of chromosomes, but the basic difference from the first year we have studied, prokaryotes are haploid, they have a single circular DNA as their chromosome, and but eukaryotes have linear chromosomes with two termini, and the difference between prokaryotic and eukaryotic chromosome is that prokaryotic DNA is said to be naked without association of basic histone proteins, but in contrast eukaryotic chromosomes are linear, they are having the association of basic histone proteins for their organization. So, this is the major difference between the prokaryotic and eukaryotic chromosome, but this session we are going to discuss the organization of DNA in chromosome, because chromosomes are made of two basic molecules. They are DNA, which is a polymer, and proteins. By weight, any eukaryotic chromosome consists of 1/3 DNA and 2/3 proteins. The proteins in chromosome come in two forms. They are the basic histone proteins and non-histone proteins. Basic histone proteins are associated with DNA to form the chromosome, but non-histone proteins, as we will see, they are involved in the compaction of packaging of the DNA into chromosomes. Further, they have the regulatory role in gene expression and other things, but our topic is how the chromosomes in prokaryotes and eukaryotes are organized. Let us come to this. Now, in this slide what I have shown, this is a circular DNA. In its circumference it measures about 350 micrometer, but when it is folded, around 40 to 50 loops are there, it gives rise to 30 micrometer circumference, and subsequently when each loop is further supercoiled, its diameter comes to around 2 micrometer. What I mean to say, bacterium is a small cell. If you look at E. coli, E. coli is a rod, E. coli is a rod, its length is around 10 micrometer, but its diameter is around 2 micrometer. We know the bacterial chromosome is present as a nucleoid in the central region of the bacterial cell. Okay. So, with this 2 micrometer diameter cell, how the long DNA, which is assuming a circle, if we uncircle this, if we take the length of that, it comes around 1100 micrometer, 1100 micrometer. So, 1100 micrometer long DNA, how it is packaged into a short bacterial cell which has a 2 micrometer diameter. Definitely DNA is, as we know, it is a negatively charged molecule, it is acidic. The phosphates present in the sugar phosphate backbone imparts or gives negative charges. Very often we say DNA is polyanionic, it has many number of negative charges in its backbone. How? This circular DNA also having this negative charges in it, can it be packed into the small bacterial cell? So, here from this we have to say that it is folded, and in each loop the DNA is supercoiled just like the telephone wire or telephone receiver cord. So, for this compaction histones are not involved, but polyamines, small proteins having positive charges in them, they are called polyamines, especially spermine and spermidine. Spermine and spermidine along with RNA are involved in compaction of this circular DNA into a supercoiled folded chromosome. So, without discussing about the DNA of E. coli cells, I thought it would be incomplete in teaching organization of DNA in chromosome. Next comes the eukaryotic organisms, eukaryotic cells. They are more complex. DNA is present in much higher amount in eukaryotic organisms. So, if we think of human chromosomes, we have 2n is equal to 46. The chromosomes are numbered from 1 to 23. Chromosome 1 is the longest chromosome, and chromosome 22nd and 23rd chromosome, that is why they are the shortest ones. What we know, the chromosome number 1, chromosome 1, it is having nearly 10 centimeter of DNA in it, around 10 centimeter DNA. If all the DNA molecules from all the chromosomes are taken and laid end to end, joined end to end, this would come above 2 meter. Sometimes it is written 2.2 meters, or in general perception we can say more than 6 feet long DNA is present in each eukaryotic cells, spread over 46 DNA molecules of different length. Chromosome 1 has 10 centimeter long DNA, but my question is how this 2.2 meter long DNA is present in each nucleus, which is below 10 micrometer. The nucleus of each human cell comes below 10 micrometer. How this small nucleus houses 46 pieces of DNA molecules present as chromosomes, and as we know each DNA irrespective of its base sequence has negative charges in its surface because of the presence of phosphates in it. Phosphates impact negative charges to this. So, now question arises, how this long DNA can be processed, packaged into small chromosomes. One aspect I want to tell you here, just for a comparison, how much long DNA we have in our body. Each cell around 2.2 meter, trillions of cells we have in our body. It is enough to travel with this DNA from earth surface to solar surface, likewise 300 times. It is amazing. So, definitely we are the products of evolution. DNA is packaged along with histone proteins in natural course. We have not done this. So, therefore, it is nature which has packaged the entire gamut of DNA in the nucleus into chromosomes. Nature is an expert packer. So, let us have a basic fundamental how this DNA having many negative charges can or is packaged into small chromosomes. So, now it is DNA along with histones, which are basic histone proteins. Histones means they are basic histone proteins. There are two different types of histones. So, basic histone proteins along with the DNA, they play important role in packaging. Now, I can say you with a model how these histone proteins help in packaging the DNA. Now, in 1974 it was observed that histone proteins are regularly associated with the DNA to form nucleosome fibers. Now, I want to say you this ball is a histone core which consists of 8 histone proteins, and DNA which has negative charge, it is wrapped around the DNA in a left handed manner to form a nucleosome. Likewise, at regular intervals DNA is wrapped tightly around the histone cores to form nucleosome. So, the basic fundamental unit of eukaryotic chromosomes is the nucleosome fiber, and these beads are nucleosomes, and these beads are interconnected by the DNA, and DNA is continuous. So, the DNA in between two nucleosomes is the linker DNA. So, now I want to tell you each histone core is a histone octamer. It is a disc shaped structure consisting of 8 histone proteins, 2 molecules of H2A, 2 of H2B, 2 of H3 and 2 of H4. They form the histone core, and around which the DNA, the negatively charged DNA is wrapped in a left handed manner in 2 complete turns. The point of entry of the DNA around the core and the point of exit, they are sealed by one extra nucleosomal protein called H1 protein. So, this is a complete nucleosome. So, what is seen, what long of a DNA is wrapped around this? It is around 166 base pairs of long DNA is wrapped around this histone core to form a nucleosome. So, the linkers, they vary in length, they vary from up to 50 nucleotides long. Now, I want to say that in the cell, chromosomes and this chromatin, there are two terms. So, chromosomes where they are present? Now, chromosomes are present in non-mitotic cell. Chromosome non-mitotic cell, sorry, it is present in mitotic cell. That means, a cell dividing has chromosomes, but non-mitotic cell, that means the cell which does not divide, that is in interphase, there the chromosome remains as chromatin. So, chromatin is extended form and chromosome is condensed form. So, this is by way of condensation, give rise to chromosome, and chromosomes become distinct during the time of division, that is during mitosis or meiosis. But this chromosome, as the cell division is over, they undergo decondensation and become chromatin. So, in the cell cycle at regular intervals, on the completion of the cell cycle, completion of division, what exactly happens? The chromosomes decondense to form chromatins, and again during cell division they undergo condensation. Therefore, chromatin exists in this chromatin exists during interphase stage. So, now this chromatin, when revealed under electron microscope, chromatin usually exists in 30 nanometer thick chromatin, and these are called solenoid, but when they are further extended, it gives rise to the beads on a string appearance, which is a nucleosome fiber. So, the nucleosome fiber is the basic level of organization of chromosomes, and that further condenses to form solenoid, then scaffold, then it gives rise to the mitotic chromosome, mitotic metaphase chromosome as seen from this diagram. Easily you can understand how the DNA is packaged into higher order organization to give rise to the mitotic chromosome that divides. So, this chromatin exists as two forms, euchromatin and heterochromatin. Euchromatin is light staining chromatin, and that is accessible to DNA polymerase, RNA polymerase for gene expression. So, euchromatin exists in cell around 90%, but very rarely around 10% of the chromatin exists as heterochromatin, differentially thick, differentially staining chromatin. Heterochromatin is usually not accessible by cell enzymatic system for expression. So, heterochromatin usually non-expressing, but euchromatin expresses, or euchromatin is accessed by enzymes of cellular system for transcription and replication as well as different activities. Before the proof of DNA as genetic material, it was thought that any molecule that would be acting as genetic material, whether DNA or protein, must have faithful reproduction. It must have the property to replicate, but with the discovery of a DNA double helix, there are two strands, Watson strand and Crick strand, the two strands in which the bases are complementary, and Watson and Crick's proposition was that the complementarity of the bases of one strand to that of the another would be the key to faithful production of DNA or replication. So, even though they did not know how genetic material replicates, but they proposed that this complementarity of the bases would be a key to the replication mechanism or replication procedure. So, complementarity is essential for either replication or transcription. So, now why replication should take place? We know when a cell divides, it gives rise to two identical cells, whether it is bacterium or the human cell or plant cell, because a cell divides mitotically to produce two identical cells. Therefore, the genetic material which is DNA, that must be faithfully copied and segregated to two different cells called daughter cells. From this diagram, if you analyze the chromosome, two chromosomes shown here, they are in the interphase, in the S phase that undergoes replication. In each chromosome, there was one DNA before S phase, but after S phase, each chromosome has two chromatids, two DNA molecules, and then the two DNA molecules in the form of two chromatids which become distinct at metaphase stage, they are separated into two different poles. So, DNA replication is must to take place before a cell divides. Now, how this replication takes place? What is the mode? So, after 1953, different scientists were engaged to prove how this DNA replication takes place. So, in that line, Michelson and Stahl of California Institute of Technology, they proposed the semi-conservative mode of replication, which was this was emanate from the Watson and Crick's view, but side by side some other people they also proposed conservative mode and dispersive mode, conservative mode, dispersive mode and semi-conservative mode, but the first two are not proved, but this semi-conservative mode of DNA replication was proved in E. coli by Michelson and Stahl, what they did, now they took E. coli, and they grew these E. coli cells in a culture vessel, with N15 nitrogen, ammonium chloride. N15 ammonium chloride was the sole source of nitrogen. When they grew for several generations, the bacteria, they took up this ammonium chloride and incorporated in all proteins and DNA, because bases in which nitrogen is present. After growing for several generations in ammonium chloride, N15 ammonium chloride, which is heavy in comparison to the normal N14 nitrogen. So, this differs by 1 neutron only, but when this difference is taken into account in a whole DNA molecule, so many number of nitrogen atoms are there, definitely the DNA having N15 nitrogen would be heavier than N14 nitrogen. So, what was their basis of experiment? They grew this bacteria E. coli for several generations, so that all the bases have N15 nitrogen, and then what they did, they transferred all the E. coli cells to a new culture medium in which there is N14 but normal nitrogen. After this, when they transferred, they wash these E. coli cells, because if any contamination of this ammonium chloride is there, to avoid that they transferred this, and subsequently they allowed them to grow, took the samples at intervals of 20 minutes, because E. coli reproduces every 20 minutes. So, they took the sample at 20-minute intervals, first 20 minutes, then 40 minutes, then 60 minutes, means first generation, second generation and third generation. Then they centrifuged, they extracted the DNA, centrifuged this. But my question is, the basis of their experiment is that N15 DNA in a centrifuge tube would acquire this in the bottom, but N14 nitrogen, N14 DNA will be light DNA, and this is N14, and this is N15 nitrogen, this is a heavy DNA. But when they transferred from N15 to N14 in the first generation, they found a band intermediate between the two heavy and light. So, this is hybrid. By this basis, how they could differentiate between this heavy, light and intermediate hybrid DNA, which has both N15 and N14 in their DNA. So, this was the DNAs were subjected to equilibrium density gradient centrifugation developed by them. Okay. So, now what I want to tell, they transferred the DNA, they extracted the DNA at intervals from these E. coli cells, subjected them to density gradient centrifugation, and they found that in the first generation the DNA occupied an intermediate position. As we see this diagram, in the first generation the DNA occupies an intermediate position, suggesting that it is having both N15 and N14. But in second generation, if you see, in second generation 4 DNA molecules will be produced. If you look at this diagram, it will be clear cut. So, out of 4 DNA molecules in 4 cells, okay, 2 would be completely light DNA and 2 would be of intermediate. So, heavy, no heavy does not appear in second one, only intermediate DNA by hybrid DNA and light DNA. As they grew for several generations, like 3 generations, 80 DNA molecules would formed, 4 generations 16 molecules would be formed, but in 16 out of 16 DNA molecules after 4 generations, what is seen that only 2 DNA molecules will be of intermediate position. They are hybrid, but rest are light. So, their definitive experiment proved that DNA replication is semi-conservative. As we come to this slide, you can see this. There are 2 strands, the 2 strands separate, and seeing the basis of one strand which acts as template, a new strand is formed. So, from one parent DNA molecule, 2 daughter DNA molecules are formed. In each daughter DNA molecule, one is template DNA which is parental, and the nascent strand or new strand is there. So, as a result of which, in each daughter DNA molecule, one is parental strand, the other is nascent strand or new strand. So, therefore, 50% of the parental strand or 50% of the parent DNA is conserved in the new one. So, that is why it is called semi-conservative mode of DNA replication. So, in E. coli, this semi-conservative mode of DNA replication was proved by Michelson and Stoll in 1958, and that was subsequently also proved by Taylor and his group in Vicia faba, which is a eukaryotic system. They used tritiated thymine to label their DNA, and then they went for or came for autoradiography. By autoradiography technique in Vicia faba root culture, they found the same thing happens, DNA replicates semi-conservatively, and further this was also proved in mammalian cells. So, in eukaryotic cells and prior to that in prokaryotic bacterial cells followed by eukaryotic cells, DNA replication was proved to be semi-conservative, in which in each daughter strand new molecule, one is parental, one is nascent. So, the 2 other modes proposed, that is conservative and dispersive modes, could not be proved. So, now further we will see how this semi-conservative DNA replication takes place at molecular level in E. coli cells. DNA replication involves 3 basic steps. As we know, prokaryotic DNA, prokaryotic chromosome consists of a single circular DNA, that is double stranded circular DNA. Question arises, the 2 strands or the 2 strands, they unwind as a whole at a time, then replication occurs, or replication takes place at specific points. It is seen that replication takes place at specific sites, specific regions, and from there it proceeds to rest of the DNA. So, now DNA replication proceeds at specific points in chromosomes called points of origin or points of replication. So, bacterial chromosome has a single replication point from where replication starts. It is specific, anywhere it cannot start with. So, now whether it is prokaryotic chromosome or eukaryotic chromosome, all of them follow 3 basic fundamental steps. What are these? The 2 strands separate, because in a double helix the 2 strands are opposed to each other and the bases are not exposed. So, how the enzyme can access? So, therefore, the 2 strands must separate, they must unzip, they must open, so that the bases are exposed. So, unwinding is the first step, and second one is each parental strand acts as a template, to which the new bases are by way of complementarity attached, and the bases in the new strands, they are joined by the enzyme to form the new strand. And finally, in the third one, the parent strand and the new strand, it means in each daughter DNA the 2 strands wind to form the helix, and subsequently they associate with histone proteins to form chromatin. Whether it is prokaryotic or eukaryotic replication, it is a complex process. Before going to the proper mechanism, let us have an idea about the components involved in. The major components of a DNA replication or requirements for DNA replication we discussed. One is the template strand that is available by unwinding of the two strands. Second, the nucleotides, the building blocks. The building blocks are deoxyribonucleoside triphosphates. Deoxyribonucleoside triphosphates are the building blocks. So, DATP, GTP, DCTP, DTTP, they are the building blocks. Okay. As they are the triphosphates, they are not only the building blocks as well as they are the energy source, because during polymerization each bond formation, nucleotide to nucleotide bond formation involves energy. So, the triphosphates are the source of energy as well as the building blocks. But who will do this polymerization? By way of complementarity, the bases will attached, A, then T will bind, C, G will bind, but what will form the bond in between? Enzyme is necessary. So, the enzyme necessary for this polymerization are DNA polymerases. So, 3 DNA polymerases have been discovered in the series. The first one was discovered by Arthur Kornberg in 1958. Subsequently, another 3 DNA polymerases have been discovered, and they are named in order of their discovery DNA polymerase 1, 2 and 3. They are given the Roman numerals 1, 2 and 3. So, we discussed the the building blocks, the nucleoside triphosphates and the enzymes, the 3 enzymes DNA polymerase 1, 2 and 3. But we must understand the property of each enzyme, each polymerase. But I want to say, out of the 3 polymerases, DNA polymerase 3 is most important. It is the most important polymerizing enzyme. The basic property of this polymerizing enzyme is it cannot start a chain new. That means it cannot initiate a new chain. So, that is why it requires a primer. It means if a 3-OH end is available, whether it is of DNA or RNA, if this end is available, from there it elongates. So, now I can say this 3-OH end in this replication process is provided by RNA primer. So, RNA primer provides this 3-OH end from which the polymerase elongates. Second one, this DNA polymerase 3, DNA pol 3, what we learnt, it always polymerizes from 5' to 3' direction. The direction of polymerization is 5' to 3'. So, that is why always an arrowhead is given at the 3' end. So, it is the growing end, and further it is also bifunctional. It has proofreading, it has proofreading mechanism. It goes for polymerization, it adds new nucleotides to the growing end, but whenever there is any mistake or a wrong base or nucleotide is incorporated, then it comes back, excise, ताको काटी दे, तापर बाहर कर दे, then another new base is incorporated. So, फेरी आसिकी काटिकी से जो wrong base ताको remove करी, again the new base is incorporated. That property is called proofreading property. So, DNA polymerase 3 has polymerization property, polymerizing activity as well as proofreading activity. Okay. Proofreading activity. So, my discussion on this is that this major polymerizing enzyme needs a primer for elongation, and that primer is provided by RNA primer. Okay. It cannot in any cost grow in 3' to 5' direction, it is always from 5' to 3' direction. So, now what we have to discuss that, as you know, E. coli has double stranded circular DNA as its chromosome. The replication takes place at a particular point, that point is called ORI-R, origin of replication. There the enzyme helicase binds, unwinds the two strands by melting the hydrogen bonds, because the two strands are held with each other by means of hydrogen bonds between the bases. So, helicase dissolves the hydrogen bonds and thereby the two strands get separated. If you look at this diagram, and this is the chromosome having double stranded DNA, and this point is ORI-R, origin of replication. There the helicase binds and the bonds, hydrogen bonds dissolve and they separate. Thereby they create an eye or bubble. So, it should be kept in mind the two strands entirely do not break apart, only locally there is base pairing, only locally there is dissolution of hydrogen bonds, so that an eye or replication bubble is created. Now, here the two strands separate. Now, each strand acts as template, and from there DNA replication proceeds, and what we see, DNA replication is bidirectional. Here in this direction replication proceeds, and in this direction replication proceeds. So, replication proceeds from this point and proceeds in both the directions, and ultimately they meet at the other point. Now, what I can say, this replication point or replication bubble or eye, where there are two forks. So, if I divide this can be a fork and this can be a fork. The same thing happens in eukaryotes. So, now at each replication fork, each replication bubble, there are two forks, and we will discuss what is the molecular mechanism of a DNA replication at each fork now with the help of a diagram. E. coli chromosome consists of one replicon. Replicon means replication unit. From the point where replication begins and the termini, the ends where the replication ends, that is called a replication unit or replicon. So, E. coli chromosome is a single replicon. Now, what we see, there is a single point of origin, and from there the replication proceeds bidirectionally, and there are two forks. Now, we will see how, what are the events, what are the mechanisms by which this replication takes place at a molecular level, because DNA replication is coming as a long question in our course, in our curriculum. So, what we see, let me draw the structure of a replication fork. Now, this is one strand of DNA, this is another strand of the DNA. We will discuss. So, now this is 3' end, this is 5' end, because DNA is antiparallel. DNA is antiparallel. So, how the two strands are separated, what protein is involved? We will see. The helicase is a protein, helicase, helix unwinding protein. Let me tell you, this is helicase. Helicase breaks with the help of ATP, breaks the bond, and the fork moves in this direction. So, this is the direction of the fork movement, and now these are the two template strands available. We will see how the new strands, nascent strands are synthesized by the enzymes and the proteins involved. So, now we discussed this enzyme needs a primer. This is a primer. This is a short segment of RNA. This is synthesized by enzyme primase. So, this is primer. Primer provides a growing tip, a 3'OH end, that is all. And from there the DNA polymerase 3 takes on, it elongates like this. I draw a line like this, but it is not like that. If I draw, here is A, then T comes, thymine nucleotide comes, C, guanine nucleotides comes. Likewise, by way of polymerization, this chain elongates, and this is the 3'OH end. This is the 3'OH end, and DNA polymerase 3 only elongates in 5' to 3' direction. If you look at the opposite strand, opposite template strand, this runs in this direction. How that can be? Here it will be problematic. We cannot draw like this. The DNA new strand cannot be synthesized in this direction, because here lies the 3' end. So, therefore, in the other strand the problem is something different. Here in one template strand which runs in 3' to 5' direction, the new strand is synthesized from 5' to 3'. It is always the case by the enzyme DNA pol 3. Now, I want to mention the other strand, how it is synthesized. How it is synthesized? It is not synthesized continuously, it is discontinuously synthesized. It is synthesized as patches, as small segments. Then these small segments are subsequently joined by other enzymes. This is very interesting. Similarly, here we can see here one RNA primer. To this RNA primer, the DNA elongation takes place here, but prior to that another enzyme is another segment is there. Here it is primer, it is the primer. What I mean to say, in the discontinuously synthesized strand, small segments of nearly 2000 nucleotides are synthesized, and in each segment, which is called Okazaki fragment, because this Okazaki fragment was discovered by Reiji Okazaki of Nagoya University of Japan. He by radio labeling technique, by tracing technique, he found that new strand is synthesized as patches, and subsequently they are joined together to make it continuous. How this happens? So, now this is a primer, and this is the 3'OH end of the DNA. Now, this is the 5' end, 5' end. So, here the enzyme DNA ligase, as we know, we have read this, DNA ligase ligases this, joins this and makes continuous, but here I want to mention the enzyme involved here. Let us come to this point. It will be very clear from this diagram. Here this is the primer, and from that primer DNA polymerase 3 elongates up to this direction. So, now can this segment be joined to this segment, because this segment has DNA here, it is DNA, and this is RNA, this is DNA. So, DNA RNA cannot be like that, it cannot be patchy, it must be DNA strand. So, now another enzyme which is called DNA pol 1, DNA polymerase 1 comes into action. It is a bifunctional enzyme, it is an editing enzyme, it edits. What it does? In one hand, when this enzyme comes into force, it removes the RNA primer. This DNA polymerase is a 5'3' exonuclease, 5'3' exonuclease, and side by side it is also 5'3' polymerase. So, these two it is a bifunctional enzyme. The dual action of this enzyme, what it does? Now, it removes the RNA primer by 5' exonuclease activity, and side by side it elongates by adding new nucleotides to this 3' OH end of this DNA, so that the gap is filled. So, now after some time this RNA is removed, and side by side this DNA is elongated, and as a whole this RNA is removed. So, now there are two termini, 3' end and 5' end. Now comes the enzyme DNA ligase here. So, this is the mechanism by which the short segments are joined by enzyme DNA ligase, which is ATP dependent. So, my question is, one strand is synthesized continuously, the other strand is synthesized discontinuously. So, this kind of replication is called semi discontinuous. DNA replication is semi discontinuous, and priorly we have read semi discontinuous. Before that we have also read it is semi conservative. So, DNA replication is both semi conservative and semi discontinuous. We know now this is a DNA molecule. In this is the parental strand and this is the nascent strand. This is parental strand, this is nascent strand, new strand. So, 50% in each DNA, in each daughter molecule, one strand is parental, one is new or nascent, and this is continuously synthesized, this is discontinuously synthesized, and subsequently after removal of the RNA primers, they are joined by DNA ligase, making this continuous. So, with direction of the fork movement, this goes on. Now, I can tell you this continuously synthesized strand is often written as continuously synthesized strand, leading strand. This continuous strand is called leading strand, and this discontinuous strand is called lagging strand. What is the meaning behind? Because 3' OH end is growing end, as this 3' OH end of this strand is much ahead of the 3' OH end of the discontinuously synthesized strand. So, that is why this continuous strand is called leading strand and this is called lagging strand. Okay. So, continuous, discontinuous, leading, lagging, it is clear. When this fork opens or this fork moves, it is seen that the helix is tangled ahead, agare gude hi jauthibo. It will be difficult for the enzyme to move. So, therefore, the tangling is removed by action of the enzyme topoisomerase or called gyrase. So, gyrase or topoisomerase 2 or gyrase is the enzyme which removes the supercoils created ahead, so that the fork can. So, as a result of which the DNA replication mechanism is very complex process, and DNA polymerase 3 goes for polymerization, DNA polymerase 1 goes for editing, it removes the RNA primer and then goes for DNA ligase goes for joining the two segments, making that continuous, as a result of which as the fork moves, the two strands, the new strands also move or new strands are synthesized. Let me mention DNA replication is a local event. The entire DNA is not opened off and then synthesized. It is by way of formation of replication bubble or replication eye, from where DNA replication proceeds bidirectionally. And now I want to mention whether prokaryotic DNA and eukaryotic DNA have the same mechanism. Basically, the mechanism of DNA replication in prokaryotes and eukaryotes are the same, but prokaryotic DNA or circular DNA is 1100 micrometer or just 1 millimeter, but our DNA chromosome 1 is almost 10 centimeter. If DNA replication starts from one point and proceeds to the other, it will take several days. Therefore, DNA replication in eukaryotes starts at many points, or otherwise to say, in other words I can say DNA in chromosome has many replication units. So that replication in, you can look at this slide, O stands for origin of replication and T stands for the termini, where the termination takes place. So, now from one point replication starts, it proceeds bidirectionally. Similarly, at the other point or other replicant it can. So, likewise thousands and thousands of replication unit or replicants are there in eukaryotes to augment or to help the long DNA molecules to be replicated in very short time. In human cell, this DNA replication in S phase takes almost 8 hours due to presence of thousands of replication units or replicant. But if this would start from one point in the chromosome and proceed in the other direction, it will take several days. So, therefore, the advantage of multiple replicants in eukaryotes is to finish the DNA replication in stipulated time. And another major difference of eukaryotic DNA replication from prokaryotic DNA replication is that prokaryotic DNA replication is almost 2000 nucleotides per second. That means, DNA polymerase 3 is very fast. It polymerizes 2000 nucleotides per second. In contrast, when it goes for fast polymerization, there is possibility of incorporation of wrong bases or error may occur, mutation may take place. They have proofreading mechanism by way of their 3-5' exonuclease activity, but on the contrary eukaryotic DNA replication is very slow. They add around 50 nucleotides per second. Because eukaryotes are multicellular organisms, mistakes cannot be dispensed with. So, mistakes should be at the minimum level. Therefore, to cope with slow rate of DNA replication, eukaryotes have multiple replication units or replicant. Here I can say replication is bidirectional, because from each point replication proceeds in both the directions. From one replication unit it proceeds and it meets the boundary of the other replicant, as a result of which the entire eukaryotic chromosome is replicated. Okay. Now, replication must be faithful. In spite of this, there are mutations. So, during replication mutations occur, but this is minimized due to the proofreading activity of the DNA polymerases and the cellular system. Thank you.